So, so thank you, Dr. Invakandan sir, for giving me a wonderful opportunity for creating such a wonderful platform to describe about our potent applications of marine resources. So thank you, sir. So we will straight away move to our today's second session, that is Seaweeds, a marine treasure. So in the brochure, I know it was written as a potent bioresource. Thus, seaweeds, they are the untapped potent bioresource so far. Still, there are several works have been continuously going on throughout the globe. So, as one of the main work, as in one of our earlier session, Dr. Mohammad Riaz has I was narrating about his experience on the ISS disease, because this ISS disease, it caused a very tremendous economic loss, not only in India, throughout the Southeast Asian countries, because most of the countries, they are depending upon the cultivation of seaweeds. There in their country, they were using for several food and as well as other applications. Because of this bacterial disease, the entire southeast coast seaweed cultivation has come down. Okay. So even that PepsiCo and Aquagri, they transfer these seaweeds and preserved in the Gujarat coast. So that is the most important information in continues to its presentation. So it is very important, because during 2014 and 15, even we got affected, okay, in our area where we cultivated these seaweeds. Okay, so one of the question also was asked whether in any other seaweed species also it was found. In certain species, not only in caraginophyites, even in alginophytes also we could see some of the a kind of bacterial disease was affected during that period, 2014 and 15. So that is an interesting thing we have to look into further. Okay. So now we will move to the other part, that is the what are the most important aspects of seaweeds. Okay. So we all know that our marine ecosystem is much diversified, starting from deep oceans to coastal reefs, from mudflats to the seagrass beds, then up to the coastal wetlands. So yes, they are providing a maximum essential services like food, climate, oil and gas, the minerals, then carbon capture for climate change, then as also as a renewable energy. So because of these uh, potent activity, bioactivity, so this UN also has given, that is we have to conserve and we have to sustainably use the oceans, seas and other related products under the Sustainable Development Goal 14. So that is our motto of this 2-week long webinar series. So we have to conserve our marine bioresources, because we should not over exploit, or we should not uh, deploy it from our natural habitat. So that is our aim to conduct this kind of webinar. So in India, we have a very good coastal line, that is almost 7,500 plus kilometer is there, and we in East Coast we have very good exclusive economic zone. Then in West Coast it was high than the East Coast. Then we are also having some potent pristine islands are also there, like Andaman, Nicobar Islands and Lakshadweep Islands, where much pollution is being there. Okay, so this makes to concentrate, because even in Niti Aayog in our last year in 2020, they have declared that we have to go for massive seaweed cultivation, because due to their high potential value, we have to go for it, because they provide a sustainable livelihood development for coastal wetlands and coastal livelihood. Okay, so in India, we have a very good species of diversity in marine resources, starting from higher invertebrates to fish, then algae. So algae found to be the third highest biodiversified organism in India. So this includes both micro and macro algae. So these are the potent and economically value species which are quoted here. So apart from phytoplankton, zooplankton, and marine fungi, bacteria, the mangrove vegetation, amphibians, mammals, and seagrass. So this several marine natural products has been narrated by our Dr. Masala Mani sir in 2 days before session. So it was also it continues to that. I'll be concentrating only on the seaweeds. So because seaweeds, we all know that they are the macroscopic or macro algae. Okay. So they are multicellular and they are widely used for food in Southeast Asian countries, Europe and US, North American and South America and all. They are continuously using for this food and medicine, cosmetics and other applications. Whereas in India, we slowly started for extraction of Phyco-colloids. So Phyco-colloids are nothing but they are the alginate, carrageenan, and agar, which are being extracted from the brown seaweeds, red seaweeds, as well as other form of seaweeds. Okay, so in India, we can see a large diversified group of organisms. Particularly, you can see here in the southeast coast, that is in Mandapam region, both in Park Strait as well as Park Bay. Okay, so we can see around 302 vulnerable economical species are being found here in this region, and they are found to be the very protected area because there are protected island like 21 islands are there. So in that region, we could not collect a single bioresource without proper permission from the environmental department. Okay. So next to this Mandamam region, we could see in Gulf of Kutch, that is in the Gujarat coast, we can have a very good number of seaweed diversify is there. Then next to that is the Malabar, that is Konkan coast and Maharashtra coast. You can see almost 152 species. Then apart from that, in Lakshadweep and near to the Andhra Pradesh, then as well as in the Andaman Islands. So these are some of the very good species based on their family and their type of species is being widely distributed in both the coasts. So because of all its potent bioactive compounds and other high value byproducts are being developed from seaweeds, why don't we call it as a sea vegetables? Because this was debate is going on whether to replace the seaweeds into sea vegetables. So very soon they will be call it as sea vegetables because they are vegetarian in nature. So as well as they are highly fortified with minerals. Almost all the half of the table of the periodic table can be seen in this seaweed Phyco saps. Okay, so in China, they are the foremost to use the seaweeds in their traditional medicine. So they date back before 3000 BC, and they were also using it as a food and in cosmetics and in other industrial applications. So in highly it was confined both in the East Asian countries and Europe. East Asian country means like Indonesia, Malaysia, Thailand, then Japan. They are also sometimes they'll be consuming it as a salad. So before soup, they are consuming it. In India, they slowly some of the edible species are converted into food products, and again we are started consuming it. Because of this liberalization, several spas have been evolved, like the Thai spa, and where we can see the soups made out of from these seaweeds. So before that, it was exploited only for extraction of the Phyco-colloids, that is carrageenan and alginate as well as other. So this PepsiCo are the leaders, pioneers in cultivating the seaweed cultivation in the southeast coast of Mandapam region up to Tondi, Pudukkottai district, up to Pudukkottai. So where we can see an enormous business model for self-help group development. So they have been continuously cultivating and they are getting the biomass as such. So at the rate of 11 to 15 rupees. Then if it is a dried biomass, they'll be giving a economical value of up to 25 rupees depending upon its quality. So this has been, they have been transfer and exported, and it will be processed in several areas like in Manamadurai, in Ranipet, as well as in Gujarat. So these are some of the region. Later, this PepsiCo has transferred this technology to AquaAgri. So this AquaAgri has acquired this technology, and again they have cultivating in the both the east, southeast coast as well as western coast, particularly in Gujarat. So they are continuously obtaining the biomass, they are processing, and they are going for export of the uh, value-added products from it. So in India, this CSMCRI, that is Central Salt Marine Chemicals Research Institute in Bhavnagar, Gujarat. So they are the pioneer in the seaweed research. So they have developed and modified several methods of cultivating the seaweeds, uh, both by monoline culture as well as the raft method. So these are the two prominent method used for cultivating the seaweeds as well as the other form of edible seaweeds. So they usually grow in shallow seawater. So where there is less sea roughness. So like area like in Pulicat, then in Mandavam region, where there is much no wavy action, there we can cultivate these seaweeds. So they abundantly grow. That too, this Kappa ficus, Gracelaria, Gelidia, then even certain sargassum species are being cultivated in this region, and they are harvested for their potent value added product extraction. So this has been commercially important for not only for food, not only for its phyco-colloids. There are several drug carrier materials have been extracted. Pharma will be seeing one by one in the later slides. So we have extracted its sap for its application as a liquid biofertilizer and paste. So these are the two products we have developed from, and it has been commercialized. The name JPR biofertilizer. Then, as I said, PepsiCo has transferred this technology to AquaAgri. Then we have for extracting the seaweed sap, phycosap, we have our own indigenous pulverizer, so which helps to extract the liquid part of the phycosap from the seaweeds. So for that we have patented and we have got the grant also. So since this seaweeds extract is highly enriched with macro and micronutrients, starting from nitrogen, phosphorus, potassium. So this they are very much high in potassium fertilizer level. So this is very well used for as a potent biofertilizer. So almost many companies have evolved from for making this fertilizer. So like companies like Snap, C6, then uh, Bharadam. So there are several biotech companies are being involved for extraction and is commercially selling as a biofertilizer. So in addition to that, it contains amino acids, vitamins, plant growth regulators. So make it as a potent biofertilizer. So I have started in the year 2002 along with Dr. S. Kameshwaran, integrated farming in aquaculture ponds. So because this has the ability to scavenge the certain pollutant as well as the heavy metals. So this since it has a carboxyl group, so it easily get scavenge the heavy metals and other pollutants from the uh, aquaculture ponds. So we are utilized as it as a biofilters in monoline cultures along with the aquaculture streams. It reduces its nitrate, sulfate and phosphate so that there is no need to change the water up to 105 days. This we have achieved in the year 2002-2003. Then with the help of these three seaweeds, we have achieved it. Then at present, there is a great demand in the global market. Okay, so I have showed a data from the internet that is the by 2028 there is a huge tremendous increase in both in all the continents. So that's why so this Niti Ayog also very much interested to encourage the seaweed cultivation in our both. So again, as a food, there is a great demand. So if you are interested, definitely you can go for cultivation of these edible form of seaweeds. Okay. So we are the one of the institution which disseminating the cultivation, because continuous uh, harvesting of the or collecting the seaweeds from the natural environment, there is obviously there will be a over exploitation of harvesting as well as the there will be a very soon it will become an endangered species. So to avoid that, many institutes have identified several regions, up to 29 regions they have found out as a vulnerable thing. We are trying to make it a very good hub, seaweed cultivation hub in our around our institute, that is in Tiruvallur district in Pulikat region, as well as in Viluppuram district in Marakkanam region. So these two are very good potent area for cultivating the seaweeds. Okay. So before going into the cultivation methods, we'll see have a what is seaweed and how it looks. So many would have know about these things, but still to understand in a very good way. So I will just go very brief. That is, seaweeds are nothing but they are like a plant-like structure because they are the macroalgae without any distinguished feature of root, stem, and leaves. So even though it looks like a plant, but they don't have a very good distinguished feature of leaves or a stem or even the roots. So in this there is a modified kind of root is there, that is the hold fast. Then the stem structure is called as the stipe. There will be a small bubble like gas bladder will be there, which contains almost carbon monoxide and other gases. So they helps to float in the sea surface along with the broad and linear kind of blades. Okay. So these blades are nothing but they are the flattened portions of the thallus of the seaweeds. They have a large surface area which is necessary for the photosynthesis. Then this hold fast is the root-like structure, which most helpful to hold in the rocky substrate or any form of seashell substrates. So they are called as hold fast. Then the new nematocysts are the gas bladder, which helps to float in the sea surface. Okay, so make exposure to the sunlight for its photosynthesis. Then stipe is nothing but the stem-like region, so which connects the blade-like leafy structure and the holdfast. And what are the different forms? Based on the pigment production, they are classifying these microalgae. They are also classified as Chlorophyceae, Phaeophyceae, and Rhodophyceae. So Phaeophyceae is nothing but they contain the brown pigment, and Rhodophyceae they contains the red pigment, whereas green algae is the Chlorophyceae contains the green pigment. So almost 1,500 species are being there. This brown algae, so they are very economically viable algae. That is almost like Sargassum, Turbinaria, Padina, or some of the species we have worked here, and they are abundantly found in the Mandapam region. So to conserve and protect the over exploitation of the seaweeds, so we have disseminated this kind of seaweed cultivation to these by forming the self-help group women. So we have disseminated in two methods, that is the raft method and a monoline method. These are the standard methods which are prescribed by the Food, Agriculture and Organization, that is FAO of United Nations. So this raft method, so they're nothing but four bamboo sticks are being attached along with the diagonally and as well as in the inner region. So as a frame-like structure, then the seaweeds seeds will be tied in a rope, that is a seed rope, and the seed rope will be protected with a net. So fishing net will be connected at the below so that the seeds will be protected from the grazing animals. And this will be deployed into the sea. Okay. So this was first initiated in 2009 with the help of our Aqua Culture Foundation of India President, Dr. A. Saktivel sir. So he is disseminating this technology to us, and he was teaching a very good number of self-help group women in Muttam, and this was funded by our own philanthropist founder chairman, Dr. JPR. So he was also there. So with the help of their fund, we have disseminated in the almost 127 women. Still they are practicing this seaweed cultivation in the closed system of Colonel Dr. JPR Fishing Harbor in Muttam, Kanyakumari district. Okay. Then we almost deployed 400 rafts, and we could get almost 1 to 2 tons of seaweeds during every week of the harvest. So And next, since it was in the almost 800 kilometers away from there, so we try to disseminate this technology to even to our adjacent villages in Kanchipuram district, Tiruvallur district, as well as in Viluppuram district. So in Kanchipuram, we have transferred this technology, that is Kovalam village, Panayur, Karungatu Kuppam, then in near to Mamalapuram also we are disseminated, then in Marakanam as well as in Puliket. So since these region are high due to its high wavy action, so we are unable to deploy the raft. So this may get dismantled due to the wavy action. To avoid that, we use this monoline culture method. So with a long rope of 10 meters or 25 meters of capacity, so we will be tying the seeds along with the nets, and this will be deployed into the sea. So it is in the Kovalam region. Then in Panayur and Perikatukuppam, we have designated with the help of the local fisher folk. And in Kanathur village also we are designated the year 2015. And this is in Puliket, that is in Ponirevu village. So they are very good. Still they are doing some of the seaweed cultivation as well as other crab fattening and cage culture also they are doing along with this. So in their cage culture, they are installed these ropes for their continuous economical value. So this is the seaweed sap extractor. So to increase the economical value of the product, that is the biomass of the seaweed, what we did. So we have, because this biomass are very rigid, they are very slippery, and as well as they escapes the kind of grinding. So for that we have developed a decentralized pulverizer which is made out of a pulverizer, double pulverizer in nature, along with the milling wheels. So these are the machines which are operated with the help of 2 horsepower or 3 horsepower capacity, so that we can almost extract the phycosap from the biomass at the rate of 0.5 ton per day capacity. So this is decentralized one. So wherever we need to take to our region, like near to the seashore, we will just attach to the tractor and we can take along with the to the because they are the decentralized one. This has been applied as a patent and we got grant in the year 2020 December. From this we are extracted, because to increase its value addition, so we have extracted a biofertilizer, and this has been packed and it has been sold. Since it has almost all the macro and micronutrients along with the plant growth hormones, they are found to be a very good effective biofertilizer for all crops. They are applied as a foliar spray as well as seed treatment, soil treatment and drench method. So in certain higher altitudes, they requested to carry the enormous volume of liquid. So they found difficult. So that's why we have incorporated 2-3 seaweeds into that, and along with some of the beneficial microorganisms. So we have developed it as a paste. So this paste is being very well utilized in the higher hilly region. So this cost is around, the paste is around 300 rupees, and whereas this we are sold out at the rate of 100 to 180 rupees per liter. Okay. So these are the two products which have been developed. So after application, so we found out there is a very good increase in all the crops. So in sugarcane, we could achieve about 40 percentage of increased yield. So that is a very interesting thing. This was also proven by the CSMCRA. So they are all the one of the major CSI institute for developing and applying the seaweed products. And we have applied for our that is the traditional rice varieties in Chinnasalem, Kadalur district, Tittakudi Taluk. So where we could achieve a very good increase by 15% of increase was found to be effective in these traditional rice varieties. Apart from that, after extracting the liquid part, that is the phycosap, so we have extracted the other phyco-colloids like agar. So these are very common methods, standardized methods with still some modifications we have extracted. So like alkali acid method, so we have extracted these agar, then carrageenan. So this carrageenophyte, since they are this is, Kappa ficus and other terminaria species are very well used for carrageenan extraction. So next is the alginate. So alginate also, it is one of the very good because these are very well used in the as a food building agent and a food stabilizer. So they are highly, because in our in Jigardanda and even in Tirunelveli Alva, they used to add as a one of the main ingredient. Because of this consistency only, they give a very good uh, consist to the food product, that is the Jigardanda and the Tirunelveli Alva. So what we extracted, we get a maximum yield, because in they are the pristine region in Kanyakumari district. So where we could get the high biomass within 35 days. In other parts of the places where we have cultivated, so they go up to 45 days for Kappa ficus and 75 days for Gracilaria. But in this region, we could get a maximum yield of carrageenan and Phyco-colloids, um, then within a shorter period of time, because of it's a confluence of both the Arabian Sea as well as the Bay of Bengal. Because of that, we could get a very good growth. So that is one of the reason we find out in our place of cultivation. And this is the commercial value. Okay. So for raw seaweed, we could get around up to 15 rupees based on its quality and its content, carrageenan content. You will get a very good amount. Then for its dry seaweed, we could get up to 25 rupees. And for other thing, we will get up to 600 based on its quality. That is, if it is a refined Phyco-colloid, you will get a very good amount of so what are its production cost means? It will come around 400 rupees per 10-meter rope for all its accessories and seeds. So after 35 days to 45 days for Kappa ficus and 65 days to 75 days, we could get, we will be spending around 400 rupees per rope. Then for bamboo raft, we will get 750 rupees. Whereas the biomass we would be getting for rope, it will be around 150 kg. For bamboo raft, we will get around 250 kg. So this is a very interesting thing. So there is a very high market potential from this seaweed. So in apart from this, we have also through Atomic Energy Regulatory Board, we have carried out this project. So in which we have absorbed the radionuclides. Okay. So certain application point I am going to tell after this. So these radionuclides are being let out during emergency period or emergency preparedness. So we try to utilize these phycocolloids like alginate, carrageenan, and agar. So which helps to absorb these radionuclides. So which have been, because since it is having a high scavenging potential due to its carboxylic group present in the phycocolloid. So this alginate and carrageenan are found to be the potent uh, source to scavenge and bioadsorb the radionuclides. So this alginate and carrageenan at a ratio of 9:1 found to be a very good source. So we utilized for absorbing the thorium, strontium, and cesium also. So they also interested to work it on the iodine. That also it has been carried out, and we got a very good result from the these phycocolloids of seaweeds for radionuclide absorption. Then we can also recover the adsorbed radionuclides with the help of mild acid treatment. We could remove the whatever the adsorbed radionuclides present in the water or any form of thing. So we will be making as it as a bead like form or even a strip like thing, so that when there is a emergency, we can just apply over the exposed region, and we can during this Fukushima and all, they used certain other method of thing. So they will be isolating the people, and they will be uh, having a membrane like structure, but we can use this form of phycocolloid which used to uh, adsorb and remove the exposed radionuclides. So that is our ultimate aim of this project, which was funded by AER. So these are the some of the seaweeds which we have worked. So like sargassum species, and Gracilaria carticata, then Sargassum whitei, then Sargassum polysystem, then Padina tetrostoma. So this we are worked for extraction of these several bioactive compounds. Okay, next we are going to see about the bioactive compounds present in this seaweed biomass. Okay. So we all know that all seaweeds contain maximum dietary fibers, polysaccharides, proteins, minerals, vitamins, then pigments, lipids, and other secondary metabolites. So these are very important for making it as a edible form of macroalgae. So this we are trying to make it as one of the important edible things, so that we have extracted a certain form of seaweeds, so which are used to develop a health drink. Okay, so as it contains almost many, starting from iron, so then iodine. So there are different forms of minerals are which are used for. So we have tried to fortify this phycosap for development onto a. Still work is going on. So we are not much revealing at present, because without proper result we could not tell immediately. So we are trying to developing certain health drink from these seaweeds. Okay, then secondary metabolites, because it contains almost all phenolic very important phenolic compounds, alkaloids, then other terpenoids are there, so that we can use for several applications like antimicrobial activity. So we have worked in the antimicrobial activity of they are against certain potent nosocomial infection causing pseudomonas species. So that is very important. So because they are the multi-drug resistant variety, so certain terpenoids and alcohols are used to treat or used to show its antimicrobial activity in this nosocomial causing infection microbes. Then antioxidant activity, we have done extensive work. So with the help of ficozanthine, and we'll see one by one. Then certain anti-tumor activity also, because it activates the caspase chain. So immediately it will reduces the apoptotic activity. Then anti-diabetic. So it has also has a hypoglycemic effect. So it is very highly useful. This was very well worked by our own senior scientist, Dr. Ganesh Kumar. So he has, with the help of Sargassum whitei and other Tenerium species, so he has worked along with the nano composite. Then antidepressant activity also is found to be there. Then anti-inflammatory and anti-allergic activities also is there. Then certain species are the ability to reduce the blood pressure. Okay. So then, as I said, we have worked in ARB project. It is also used to scavenge the many heavy metals. So first and foremost thing is the carbohydrates. So we have extracted several polysaccharides as we see. So carrageenan alginate is one of the biopolymer. Then apart from that, we have also extracted this fucoidan and fucans. So from the species of Fundaria and Sargassum species, this was very well worked by our scholar, Dr. Vasant Raja. So he has made attempt to extract and study the sulfated polysaccharides from the Phaeophyceae, then as well as from Rhodophyceae, this carrageenan and agaroids. Then from Chlorophyceae, the ulvans. So these are some of the carbohydrates which are very well utilized for uh, antioxidant activity. Then from the oligosaccharides, like of oligomers of fucoidan and carrageenan, they are very well having the much potent activity of this antioxidant, anticoagulation, and immunomodulation. So this immunomodulation is very important. This can be widely used both in the aquaculture as well as in for human clinical trials. So this we have tried using one of the animal study we have tried, but still there is a challenge in applying as a immunomodulation. Still work is going on. So we are trying to find out a very good immunomodulatory agent also. Then the glucuronate, which is present in these phyco-colloid oligomers, they reduce the mitochondrial reactive oxygen species. So this we have find out in our study, this which have reduced the reactive oxygen species by these several antioxidant assays. So we will see one by one. So this is the sulfated polysaccharide. So from Sargassum tenerium and Padina gymnospora, we have comparatively studied. And so in Sargassum species, it shows much higher sulfated polysaccharide, so in which they contain the sulfate group along with the other protein, and a certain monomers also we have extracted. So we will see one by one. So this Phicos, galactose, mannose, and arabinose are the uh, mostly predominant form of monosaccharide present in the sulfated polysaccharide extracted from the Sargassum tenerium. Okay. So then from the Padina species, the sulfated polysaccharide contains the fructose, galactose, mannose, and arabinose. So they are the very good cytotoxicity, reduce the cytotoxicity effect due to the this hydrogen peroxide in the fibroblast cell line. So this is the cell line which we have used and evaluated its antioxidant properties. So then we have also studied the apoptotic capacity by the sulfated polysaccharides. So when there is an increasing usage of the sulfated polysaccharide at 100 microgram per ml, so we could get a very good form of antioxidant protection from the live and dead cells. You can see the number of cells which has been live cells are more when increasing the concentration of sulfated polysaccharides was used. Okay. So then we have also studied its senescence activity in the fish embryo cells. So from the young ones, when you are applying with along with the sulfated polysaccharide, this beta galactosidase activity was increased. With the help of stereo microscope, we have found out this staining was comparatively more when we are using the sulfated polysaccharide. So this shows that it has some anti-senescence activity. That is sulfated polysaccharide has its potential to it helps to go for a regenerative studies also. So this is one of the inference we could achieve. And what are the metabolites? So because they are having a very good secondary metabolites, as I said, these are some of the metabolites we have identified. That is benzamide, trifluoroacetyl, benzoic acid, and trifluoroacetyl, which are responsible for its antioxidant potential and reactive oxygen species scavenging potential. So these are some of the compounds which we have purified and we have isolated, and we have also elucidated also with the help of GCMS, then as well as with NMR. Okay. So one interesting thing we found out that during new moon day there is an increase of the secondary metabolites. So that is a very interesting thing. So when compared to the other time, like during new moon day, it is more when compared to the other seasons. So then pigments. So there is a very good form of pigment. So based on the pigment only, they have been classified. So this carotenoids and beta carotene, xanthophylls, and fucoxanthin are most commonly extracted and very well studied in the from the seaweeds. So we have extracted fucoxanthin from Padina by following the different methods like ultrasonication and other form of, even with the microwave oven method also we have followed. So this ultrasonic sonication was the found to be potent method where we can get maximum fucoxanthin, and this was also published in the process biochemistry. And this is also characterized with the help of HPLC. So the purified fucoxanthin along with the standard we have elucidated its characterization of the fucoxanthin. And we have found out that it's in ascorbic acid as a standard, it shows a very good antioxidant activity. And when we are used as ABTS Radical Scavenging, it's against the Trolox, which was used as a standard. This fucoxanthin shows a very good radical scavenging activity. So these are some of the results what we have studied for its bioactive compounds from the seaweeds. So there is a very much wide scope for the studying further. So so one more antioxidant activity, that is in the fibroblast cell, so which shows that this can be used as a anti-senescence activity. then it reduces the oxidative stress in the hydrogen peroxide induced fibroblast cells. So these are some of the results we have achieved. Then what are the other commercially viable products are being used in? You can see seaweed algae anti-wrinkle face pack is there. So it's a China product. It is still available in Amazon. So other products starting from food, cosmetics, even in our Indian cosmetic industry also they started using all the sea minerals which are extracted from the seaweeds. Okay, so with this I would like to conclude. So as there is a very much potent bioactive compounds are there in seaweeds, you can one by one you can work and you can make it as a commercially viable product from these seaweeds. So this is my area of work. So you also try to bring it as a vulnerable minimum viable products from the seaweeds and make it India as a Atmanirbhar. So this is our motto. To thank you all. If you have any questions, you can ask.
Thank you, sir. The forum is now open for questions. Participants, if you have any question, you can post them in the chat box, or you can give a raise hand feature so that I can unmute you and you can directly interact with the speaker.
Stanley sir, thank you. Thank you very much for the wonderful presentation, sir.
Thank you, sir.
Sir, actually, I have one small question. Sir, apart from this Phyco-colloids, apart from this gelling agent and all, even you said it is in Jigardanda and almost everywhere where they are using the colloid substance only. In some of the other countries, I think South Asian countries, they are using seaweed in the even sushi and all they are using, as well as the seaweed, the paper-like product that is also used, and they are using seaweed-based soup also. Apart from that, if we want to use our own indigenous seaweed species as an edible vegetable, what is the criteria we have to look there? Because there is some kind of sayings or myth are there. If we eat seaweed because of the salt content, we may have the problem of renal failure and all that. Whether it is true or not, if it is not true, how we can go for such kind of edible variety of indigenous species?
Sir, actually, so what are the seaweed extract we are taking? So even though it is grown in a marine water, the salinity of the phycosap or the liquid part of the extract, so it will be very very less. So there is no much salinity. Okay, so only the other interesting minerals like iron, even calcium, then selenium. These are some of the minerals are being present in that. Okay, so by consuming it directly, it has we have to refine it. So refining according to our requirement, definitely we can make it our own indigenous seaweeds as a very good. Still, we are working. Even CSMC are also they started working in edible seaweed species. Definitely, in another 5 years, we'll be getting a very good commercial viable product from seaweeds. So as a food, so it will be going to happen because they started using in several cosmetics and because they have maintained the moisture content of the our applied area. So it maintains due to its high alginic content and other high minerals. So they maintain the moisture content of the applied area, so that still for us a health drink definitely is going to happen based on see we are trying to give it as a condition based health drink. So if you want to go for as a osteoporotic patient, we will give one particular we have to clarify accordingly the content of the phycosap. Okay, for osteoporotic patient, for diabetic patient, one health drink. So for other form of like hyper thyroidism patients. So like that, we can give as a kind of health drink for their condition based. So this can be happened, but still we need some research. So after 5 years, definitely we can give a very good product in Indian scenario I'm talking. Okay.
I'm happy that you are going to give a product as well as other institutes also going to give a product within a short period of time. Actually,
Yes,
my question is, what is your answer for people who want to work on this particular indigenous species to make it as a vegetable? So you said it have to be refined. Apart from your juice, whatever you are saying, if some people want to make some kind of a biscuit, or some people they want to use it as a curry leaf or anything, is it possible? As you said, it is a refined manner. What kind of protocol we have to follow in there?
So first thing is we have to cultivate in a very good pristine area, sir. So the area selection is very, very important. Okay. So because many places the most dumping yard is the ocean or sea. So that we have to avoid. So certain regions like in Andaman, a certain area of Mandabam region, then Rana of Kutch region, then in Lakshadweep area, we have very good water without any much pollution. Due to anthropogenic activity, there is very less pollutions are there. So in this area, we have to collect. We have to select the area which is highly pristine, that is without any contamination. So first we have to do that. Then we have to cultivate the less concentrated for cultivation, like Acanthophora is there, then certain vulva species is there, then Enteromorpha species is there. So they are very good having potential to cultivate. So this CNCMCRA has developed one method. Then we are also started with the help of sporulation method. We have started cultivating this the Chlorophyceae family, which are highly edible. So this can be done. Then we have to have a very good product development. That is also important. So we have found out a place to cultivate. Then we have to go for a better cultivation method. Then process to convert into a value-added product from the edible species. So that is also very, very important. So when these are coming together, then definitely we can have a very good successful product from the market.
Thank you, sir. Thank you. Thanks a lot.
Thank you, sir.
Audience, any questions from audience?
So I have a question just
Yeah.
out of curiosity.
Please.
Sir, you said that the seaweeds that we cultivate can be refined according to our requirement, whether it's for a cosmetic industry or for a food industry. Are there any specific guidelines that we should, comply to? Like for certain dairy products, we have the guidelines set and the rules are already there. So for seaweeds, are there any available guidelines that we can use for the food industry separately or for the cosmetic industry separately? This is what I want to know.
This is United Nations under FAO. They are having a very good manual for each sector of seaweeds. So sector of industry, they are prescribing this is the method, this is the standard, and this has to be taken care while converting it as a product. So there are a very good policies there. In India, slowly we are creating through Niti Aayog as well as with the help of CSMCR. So there are standards are there.
Thank you, sir.
Thank you.
So if there is any more questions, thank you.
Good
Thanks
evening,
for
sir.
both the Dr.
Elaine,
Yes.
ma'am, one of the ma'am is there. Elaine, ma'am.
Good evening, sir. Very, very extensive and very informative talk covering so many areas where the seaweed can be applied, especially involving the society. Thank you for that. I have three questions to you. One is regarding the seaweed culture. So is it possible that we can modify the culturing method so that we can have some cultivation possible in situ conditions? Because outdoor in the pristine environment, it is very stringent, it is very hard to get them. So biomass is what we require the most. Though it is still a partially developed or differentiated system, is it still possible that we can cultivate by culturing in the lab? That is my first question. Then second question is, you have said that secondary metabolites for this seaweeds, you have found that it is more during the new moon than the full moon. Have you found that there is any possible reasons behind that? That is my second question. And I have a third question, which is regarding the bioremediation using for the aquaculture, where you have found that more than 100 days when you were able to find that it is able to grow, as well as it was able to help to decrease the water pollution. So I wanted to know more about that also. In that aquaculture aspect, what I wanted to know is that in the earthen ponds, is it possible that we can have a substratum where we can grow this kind of species for these many number of days? Suppose for shrimp it is 100 days, then that too there will be height-wise restriction is there, then the evaporation is there. Was there water inlet was water exchange was there happening constantly or at intermittent alternate periods? So all these things I wanted to know.
Definitely. So to your first question, so this cultivation during the very challenging days in 2014-15. So in certain regions they started maintaining in polycarbonate tanks. Okay, so in a tank we can maintain with the help of like the pressure and the aeration. We could maintain and we can cultivate even in the tanks. So in a laboratory we can try with the tissue culture method is there, but still people are trying to get a very good method, but they are not succeeded until. So we are also trying with some of the species. We could get only the callus kind of thing. While when you are going to the hardening into the its natural habitat, there is very less chance of getting into a very good seaweed development. So that it's a very thing, it's a challenging thing is there. But in a tank, and we can maintain in the tanks as well as can cultivate in the tank methods, or even in the open pond methods also cultivated. So this open pond method only we have utilized for as a biofilter in the aquaculture ponds. Okay, so during that time we made 3 harvests. Okay, so up to 120 days we have maintained that pond in Marakanam region in the year 2002. So for evaporation we try to, there is a exchange of water we have maintained. So without exchange of water we could not achieve it. Only thing we can reduce the nitrate, phosphate and sulfate with the use of this seaweed. So without any other addition of any other thing, we could get a very good harvest after 120 days. So that we achieved. Then for your second question, that is in a new moon day. So it shows more. Still we could not find a reason. We can say that due to the effect of the lunar, so it may increase it along with the more wavy action. So this wavy action and as well as the effect of new moon effect, definitely there is a chance for getting increasing your secondary metabolites. This would be one of the reason I could say now. There is no proper scientific reason that we have to find out. So we have to find out any cosmic rays during that new moon day will affect. There will be some impact will be there. So like that we have to find out, that we have to study. Without that I can't. Thank you.
Sir, and regarding these secondary metabolites and different kinds of exudates, whatever is being found or being characterized from these seaweeds, there is a question of finding a pristine environment. So don't you think that each place-wise, the expression of these macroalgae will change accordingly? So suppose if we are cultivating it during this season, like a pre-monsoon season, and then afterwards, if there is a lot of influx of water, riverine water will be there. So there will be a lot of pollution load and everything will be there. So in that case, don't you think that each time when we are taking these extract, we should have a quality check or something, because the chance of some bioaccumulation of some things will be there.
Unwanted pollutant may also accumulate. So there is a chance. So this we face because the right season for cultivating the seaweed is between February to May. So beyond May, there is a chance for getting more pollutants in the water where we are cultivating. Then after June up to October, we can cultivate. So in between period, we can't cultivate in any of the area in India. Okay. So we faced this problem, as you said, there is a bioaccumulation of pollutants. So in Pulicat region, so there is 2 times during a higher wave, that is high altitude. So the entire fresh water will be coming from the seaside and enters into the this Pulicat lake. So during that time, there will be a very good purity of water there without much pollution. When there is a low tide, there is a chance for pulling off, that is expelling of the lake water into the sea. So this was there. So due to this urbanization around the Pulicat lake, so there is more pollution was there when compared to the other period. So that's why we stopped during April. So after April, we will not cultivate this seaweed cultivation up to June or July. So due to a high summer season, we have to reduce the cultivating in the open water sources.
But sir, in that case, when we come to a product-oriented experiment or a strategy, don't you think that each time doing a characterization check for the quality check, it will be, isn't it expensive? Will it be cost effective?
So that is mandate, ma'am. Because without that, we can't go for any export. So we need every time quality check. So not only for waters, but even for the product also is very important. That in many of our Indian industry, it is very lagging. That we have to concentrate. Then only we can go for export of your products.
Okay. Thank you, sir.
Thank you, ma'am. Thank you. So,
sir, there is a question from Mr. Rajamanikam, sir.
Yes.
He wants to know if the seaweeds have carbon dioxide sequestration ability.
Yes. Yeah, they are the excellent carbon sequestration. So they are a very good carbon dioxide sinking live biomass. So definitely it can be used as a carbon dioxide. They are also started for carbon sink. So we are thought of working along with one of our IIT collaborator, but still it is in the, because we need a proper that is a kind of reactors. So without a fund, we can't proceed further. But it is a very good carbon dioxide sequestration biomass. It can be done.
Thank you, sir. Any other questions from the participants?
Okay.
Thank you, sir. Thank you for your enlightening presentation.
Okay. For any further queries, there is my mail ID and my contact number is there. You can, if you need, you can post your questions. Thank you.
Yeah, participants kindly make note of the email ID and contact number in case you have doubts later on. So thank you, sir, for this wonderful presentation. I request all the